Hi friends, good morning. Today we are going to discuss about bioavailability and bioequivalence. What is bioavailability? It is the rate in which the substance or its active constituent that is delivered from a drug to our body and it becomes available for general circulation, means in blood. So, for reference, we can take IV administration which have 100% of bioavailability like direct. Why? Because that is delivered directly into the blood. So here is the definition of bioavailability. What we can write is the fraction of administered dose reaching to the systemic circulation. And what is systemic circulation? Systemic circulation means blood. And as we have discussed in previous slide, for IV it is 100%, and for non-IV it ranges from 0 to 100% depending upon the drug. And nearby there is an example like lidocaine, whose bioavailability is 35%. Why? Due to its destruction in the GI and liver metabolism. Next is fast pass effect, and here some slides I will show how bioavailability is increasing. First is dose. After that, it is destroyed in GUT. Then again, that's why it is not absorbed. Then again, it is destroyed by GUT wall. Then again, it is destroyed by liver. So it doesn't reach properly to the systemic circulations. Next, what is some for see? Now I'm going to explain some important pharmacokinetic parameters. That is AUC, Cmax, Tmax. So, what is AUC? Area under curve, which measures the extent of bioavailability, like up to what extent, up to what range the bioavailability of a drug is available into the systemic circulations. Next is Cmax. So what is Cmax? It measures both of the rate of absorption and extent of bioavailability. And what we can call it is maximum concentration of drug in systemic circulations. What is Tmax? Tmax means measure of the rate of absorption, means what amount of drug is absorbed into the systemic circulations. So simply we can tell is the time that is uh, calculated after administration of drug at which maximum absorption of drug is observed. So next is AUC. Like in previous slide, we are discussing one by one. So first is AUC, area under curve. So what is it? What is the official definition? It is a robust parameter which takes into consideration all the experimental points collected in each phase of the bioequivalence study. Means wherever we are doing any kind of bioequivalence study, then it is the basic parameter which can be considered for the experiment. And second point is AUC is the principal iteration to characterize the extent of bioavailability which access the bioequivalence. Means this can be considered as a basic parameter which can differentiate between the absorption and bioequivalence. And then third point describes that it can be applied to a single and multiple dose both, and for control release formulations. And next slide describes that what are the factors that affects bioavailability. So first is physical factors like hydrophobicity, pK, solubility, availability of drugs, size, shape, these all things. Second is drug formulations like immediate release, controlled release. Then what are the excipients that are used in the manufacturing of drugs? Okay. Uh, Third is the administration of drug, in which state it is administered. Third is gastric emptying rate. So we can tell that is GI absorption rate. Next is circadian differences, like circulation, what amount of drug that is circulated in the body, and how it is circulated into the blood. What the difference is being created that varies from drug to drug. Then uh, next is enzyme induction inhibition by drugs or foods. Like some enzymes we are giving, which can be characterized as antacids, alcohol, then grapefruits, juice, which have there is two kind of interactions. Like first is drugs, second is foods, then second is transporters, in which it's proteins. Proteins and recept. Have you ever listened about receptors? They also transfer the signal. Like that, in drug also there are some transporters, like some proteins, enzymes. Okay. Third is health of GI tract. So as the drug passed through the GI tract, so uh, GI tract uh, should be healthy. Then enzyme induction inhibition by drugs or foods. As in the previous slides, we have discussed about the enzymes. So here are some examples of some enzymes. Okay. So coming to the next slides, that is individual variations, metabolic differences. Like rate of metabolism is different depending upon the different parameters like neutral, fetal, then generic, then age, gender, diet, these all things. Then the next point is disease state. Like it can be uh, like it can be in a basic state, or it can be in initial stage, or it can be in a medium stage, or there may be insufficiency in various enzymes. So these factors may be considered. Next, coming to what is bioequivalence. So what it is mentioned that drug products are considered bioequivalent if they are pharmaceutical equivalent whose rate and extent of absorption are not satisfactorily different when administered to patients or subjects at the same molar dose under similar experimental conditions. Like many experiments are considered, but at that time many drugs are used. So there what it is calculated, the rate and extent of absorption which are not statistically different. Like different drugs, like we are taking two drugs we are taking, two kind of formulations we are making, and that is being administered to the patients. But the rate of absorption, they are not different statistically. That means it may be same or it may be almost same. So it is called bioequivalence. So it may be demonstrated through in vivo, like it may be considered. What is in vivo? It may be considered by animals or human beings. And what is in vitro? In vitro means outside, like it may be performed in the laboratories. Okay. What is it? It is mentioned by equivalence may be demonstrated through in vivo or in vitro test methods, comparative clinical trials, or pharmacodynamics. So next point is drug products are considered pharmaceutical equivalent if they contain same APIs that have same dosage form, route of administration, and are identical in strength and or concentration. Like if you are taking two drug products, then their constituents should be same, and their strength, then basic factors should be considered, like strength, then route of administration, dosage form, everything should be considered. Okay. In the second point, Sam, it is mentioned whatever I told, like release mechanism, shape, size, everything, physical and chemical factors. Okay. So here are some points that should be remembered, like same amount of same APIs, same dosage forms, same route of administrations. Then what is therapeutic equivalence? How it can be considered? Like what is the first point explains that pharmaceutical equivalent, like there every factor should be same. Then bioequivalent. The bioequivalent, like biological properties of the drug should be same. And what is third point? Approved as safe and effective. Who will approve? FDA. FDA should it should be FDA approved. A drug should be approved for administration to the patients or administration as per the guidelines of the respective countries, respective regulatory bodies. Then adequately leveled. It should be properly leveled. Then manufactured in compliance with current GMPs. So, how it should be manufactured in the industry or in a lab which is approved by GMP. The last point is therapeutic equivalents are expected to have the same clinical effect and safety profile. Like as we have discussed that it should be uh, approved by the respective bodies, then they should give a certified clinical effect and safety profile of the drug. Then there is a slide which explains about therapeutic equivalence. You can read it, and I think you will understand. If you don't understand, then you can ask me in the comment or you can mail me. Okay, thank you. If you have any kind of doubts, then we can discuss. Or personally, you can contact you. Thank you.